afternoon, everyone. I'm Eugene Lee. I'm actually from Korea. I think I'm the only domestic speaker this afternoon. So I'd like to welcome you to Korea. And we experienced really extremely hot weather until last week, warm and muggy. And I guess you guys are lucky charm. Weather today is bearable, I think. It's a great pleasure to give a talk, and I thank the organizers for the invitation. And I have to tell you that there was some miscommunication about the title of my talk. So title is quite different than announced. And I know nothing about text mining and data profiling. So if I have to give a talk with that title, I have to look for volunteers here. No volunteers. So I'll change the title. The title is EVPDIA 3.0, Quality Control to the Rescue. So I'll talk about the exosome database. So I want this to be an issue-raising and brainstorming opportunity for exosome researchers and biomedical scientists in general. And I want to start my talk with the rather provocative slide where it shows a little bit of inconvenient truth in biology. So scientists at Amgen, the big biopharma in Thousand Oaks in California, published this article about 10 years ago where they attempted to reproduce 53 landmark papers in oncology. The result was very shocking because they were able to reproduce only 6 articles. That's really depressing, you know? And it gets worse. Sorry. Do I have a...
You can use a mouse.
Oh, yeah, right.
Yep.
Thank you. So if you look at the table here, so research results published in high-impact journals, they're cited similarly whether it's reproduced or not. If you go a little bit down, impact factor lower than 20, it's really depressing because results that's not reproduced are cited 10 times, at least 10 times more. So this means bad information is propagated faster in science, just like fake news is propagated in social media. So, if you think about this is really bad, right? Many attempts were made after this paper, and result was not that promising because in many other fields, same thing happened. The highest reproducibility rate was 50%, here shown here. And if you apply that to the money spent to research, US alone, about $30 billion is wasted for unreproduced research. If you break that $30 billion, break that down, Friedman and et al. Assumes that 1/3 is due to bad reagents, 1/4 bad study design, another 1/4 bad data analysis and reporting, and 10% is due to lab protocols. Right. That being said, NIH said, I've had this, I have it enough. We have to do something about this. And they took a really revolutionary measure to this problem. In 2022, they basically said, if you get money from NIH, you have to give out whatever you have produced with that money. Specifically, they mandated that grant applicants have to submit a data management and sharing plan for any NIH-funded or conducted research that will generate scientific data And this mandate will affect 2,500 institutes and 300,000 PIs. This is quite contrary if you think about it. In 1980, US government said, whatever you produced with government money is yours and your institute property. In 1980, after 1980, for 40 years, we were thinking that our data is mine, but it is not anymore. The world is changing. So my question for today is this. Would this mandate will create a credible public database that I think is essential for researchers in any field? That's my question today, and I want you to think about it. So, I want to show you a very good example of public database in biology. It's called Human Protein Atlas. And it's funded by a Swedish program. It's a private foundation. It's called Newton Alice Wallenberg Foundation. It's like Bill Gates Foundation in US. And this is run by Royal Institute of Technology in Sweden and other Swedish institutes, plus a little bit of international collaboration. What's important in this database is they have intramural guideline for collecting data in 12 different areas. They have set rules for collecting data. So they have thousands of citations. Of course, this is a EU core site. So many of you may have heard about EVpedia, one of the database in exosome field. And EVpedia was launched in 2012 by Dr. Yong-Sun Go, who I believe is in the audience. Raise your hand. No? Oh, good. Then I can criticize. Really? No, just kidding. Oh, okay. I'll tone down. This was one of the main databases in the exosome field, and it was running well until a few years ago. It stopped running due to many minor reasons. And last year, it was reopened with the help of me and our colleagues at CRIS. And we have updated the operating system, database system, and CMS structure. And we have moved this to public cloud. That's the Korean government policy. It contains at least 1,000 high-throughput datasets, information about 700,000 molecules, including proteins, mRNAs, microRNAs, and lipids, etc. So I'll talk briefly about the history of EVpedia. Like I said, it was launched in 2012. The initial version was constructed by collecting publications on prokaryotic and eukaryotic EVs using text mining solutions, and that was followed by manual creations. So it has a different design also. It was a little bit updated, so we call it EVpedia 2.0 in 2015. The main upgrade was addition of top 100 EV proteins, and also this tool called MyEVpedia, and systematic analysis tools of EVs. That was 2015. And this is the construction flow of EVpedia. So it's quite complicated. So basically, you collect data, and you can use these analysis tools. So you can, it's basically bioinformatic tools. So you can play with your data and other people's data, publicly available. And this is the summary of EVpedia. Currently, as of January 2024, Number of members, almost 9,000. Also, accesses, almost 40 million. And number of accessed countries, 90 different countries. It contains data sets including eukaryotes and prokaryotes. And the biggest data sets is coming from proteome studies, about almost over 700,000, and also contains transcriptome data sets, which is 100,000 and for mRNA and 7,000 for microRNAs. Also, it has a little bit of lipidome data and metabolome data. This is the snapshot of what you will see if you access EVpedia. And you can select search category. You can select a prokaryote or eukaryote. You can filter data set by species, in vitro, in vivo, sample type, and sample status. If you select these and click, what you will see is the list of data sets like here. Here I have selected Homo sapiens, for example. Will speed out these data sets. And if you click a specific data set, you will see the list of molecules you want to see. It contains, for example, mRNA name, uniprot accession number, all these statistics numbers, and where it was published, basically. So it's pretty good, right? It looks really useful. If you summarize EVpedia 1.0 and 2.0, what we have observed is that even from cell culture, too many EV coagulars are identifiable, because these days you have the sensitivity of detection method very high. So problem of fractionation or purification, if you just fractionate, you will see many molecules that is not actually part of EV. This is a similar problem in subcellular organic omics. If you do proteomics with, for example, mitochondria, you see like almost 5,000 proteins. The problem is not all of them are included, not part of mitochondria. Another another point I want to point out is that it did not have any quality control system. So basically, you were collecting data sets as is. You collect data after they were produced. So you didn't have any control over how they were made, where they were made, etc. So we realized for this database to be really useful and stop spreading bad information in this field, we need a reference method or protocol-based standards, right? And this method should be well established and widely accessible to the scientist, so many people can produce data according to that method. But it has to be strict enough to keep the level of quality, and it should be, of course, approved by you guys, stakeholders. That's our conclusion for 1.0 and 2.0. So we think future biodatabases should be like this. In the past, we were collecting very small data sets. And we assumed these data sets were composed of very accurate individual data, which is not true at all. But currently, it's you produce data set. It's usually very big, and it's relatively inaccurate individual data set. It's a combination of relatively inaccurate individual data. So here I'm showing a classroom where they have 6 or 7 students compared to 100 students. Here, professors, they can teach them very carefully, like this past biology. These days, we were doing this. So we have to do something about it to produce accurate or reproducible data sets. So what I'm saying is reference method approved by field scientists. For example, sample preparation method, sample analysis method, and approved data analysis protocol for this database, for this kind of database to be useful. Here I'm showing the what NIST is doing. NIST is the American Institute, National Institute of Standards Technology, and they are actually doing something with the EV. These are five different physical methods to measure EV, and also chemical method to get the information of EV, for example, mass spectrometry, microRNA sequencing. And they have listed all these possible different methodologies to measure EV. And this is the result they have sent me. And for the size measurement, what you see is, for example, even if you use the same method, PTA, particle analysis, if it's done by different hand, one by ATCC, one by NIST, they give out very different result. And also, if you use different methodology, results are quite different too. So variation is quite high. And also, if you go into concentration, same result, the variation is even higher than 30%. So what I want to show you here is it's really hard to get a real value, true value. But if you use the same reference method, at least we are on the same page, even if it's not true value. So something to compare, that's reference. So EVPDF 3.0 is not yet constructed, but we want to implement quality control system in the database, and suggested quality system is basically tiered. For example, tier 1, the data will be produced by certified lab, and the certification by peer review in the field. A very good example is ISO 17025, and the data is produced with a certified protocol. And this tier 1, if the data is marked tier 1, that is basically reference data. We can call it reference data. For tier 2, should be produced by non-certified lab, but with certified protocol. And tier 3 is no certification at all. So current data set that we have in EVpedia is basically all tier 3. But we want to produce more tier 2 and tier 1 data set in the future, so people can researchers can use data set with confidence and input some credibility in the system. And like I said, all this certification protocol should be approved by the field scientists. For example, blood workshop by ISB, what we're doing now. And if we have resources enough, we will think about manual creation in the future. So that's all for my talk today. Thank you for listening, and I'll answer any questions. Thank you.
Mitka.
Hi. So thank you for this talk. I think it came at the exactly right time because at ISEF we were also starting to think that reproducibility, of course for reproducibility, good data and good databases are also needed. And I think it was 2 years ago at the Helsinki Blood Workshop where I think, Pia, you were the person who was really strongly for having available databases for EV community to be able to reach through the maybe a simplified system to different databases that exist and interconnect them. And this is what we're doing at ISEF. So there will be a task force for databases, and I think you have to be part of it because you have good ideas. One way how to approach the databases with these certifications, but probably there are also some other ideas. And yes, just thank you for bringing this very important topic.
Thanks for the invitation. Thank you.
Can I follow up on that quickly? I'm here.
Oh, sorry.
I don't think I could understand. So what do you envision that is going to change once you go to this EVpedia 3?
Oh, so
What are the changes that you think?
basically usability of data sets that's produced by taxpayers' money. That's very, it's very government mindset, but it's going to be good for researchers too. So if you go into any database and download any OM data, how much confidence do you have regarding that data?
No, I'm not saying, I mean, I know that's going to be very important. I'm not questioning that. I'm just wondering if you have already an idea of what differences you're going to see. Like what percentage of those proteins is going to be, reliable?
Oh, okay.
If you have started your analysis.
That's a very hard question to answer, but it comes down to the money, right? You have. So you have to really produce data set with, sort of a protocol and, certain person, only certain person can produce it, all these, requirements. It really comes down to the fund you have. So next year, we're going to start producing data. I got some fund from the Korean government. So we can talk about what kind of analyte we will first approach, things like that.
We have one question from Slido. What makes an institute or lab a certified lab for the tier system?
I really
how do we make an institute or a lab become a certified?
So we have to make our own certification. So we can compose a board or task force that will make guidelines how to make these data sets, all these requirements produced by us, not the government. Right? So that's why the consensus in the field is very important.
This is a very good point.